Welcome to the Explainer. Grab your safety goggles because today we are stepping right into the shoes of molecular detectives inside a biochemistry lab. Have you ever wondered how scientists figure out exactly what components are swimming around in a completely clear, totally featureless liquid? Well, today we're unpacking the exact methods they use to solve these microscopic mysteries. Okay, let's dive into this. The building blocks of life, amino acids and proteins. They're absolutely crucial to everything we do. Yet to the naked eye in a test tube, they are completely invisible. I mean, we can't just look at a liquid and literally see a protein floating around, right? We have to force them to reveal themselves. We do this by introducing specific chemical clues that trigger very visible, often really dramatic reactions. We're essentially interrogating these invisible molecules until they confess their identity. Here's our structured roadmap for today. We've got 1 protein detective toolkit, 2 Millon's Test, finding tyrosine, 3 Ninhydrin Test, aromatic acids, 4 Ninhydrin Test, free aminos, 5 comparing the test results, and finally 6 safety and final thoughts.
Let's jump into Section 1, the Protein Detective Toolkit, Unmasking Amino Acids.
Grounding ourselves in the science here, biochemical identification is defined as the laboratory process of identifying specific amino acids and proteins based on characteristic color reactions. Color is absolutely our primary weapon. By utilizing specialized reagents, we can force specific chemical groups within the amino acids to react, creating these unique, vivid colors. It's kind of like applying Invisible Ink Developer to a blank page. Suddenly, boom, the secret message appears in bright red, purple, or yellow. Section 2, Millon's Test, finding tyrosine, the hunt for the phenolic group. So let's walk through the actual physical procedure you'd perform at the lab bench for our first case. We are hunting for a specific molecular target called tyrosine, looking for its unique phenolic group. First, you're going to take exactly 2 milliliters of your unknown protein solution and add it to a clean test tube. Next, you introduce your interrogator, in this case, just a few drops of Millon's reagent. Finally, you apply a little bit of pressure. You place the test tube into a water bath to heat it gently. You definitely don't want to boil it aggressively. Gentle heat is the key to coaxing out this reaction. And then we get the reveal. At first, as you add the reagent, you'll observe a white precipitate forming, so the liquid gets kind of cloudy. But as that gentle heat from the water bath does its work, that white precipitate goes through this really dramatic transformation, turning a distinct, vibrant brick red. That brick red color? That's your satisfying aha moment right there. That red complex confirms beyond a shadow of a doubt that the amino acid tyrosine, with its specific phenolic group, is hiding in your sample. Moving to Section 3, the Xanthoproteic Test, aromatic acids, nitrating the aromatic ring. For this case, we're actually widening our net to hunt for aromatic amino acids. This procedure requires a slightly more complex sequence. First, you add your protein solution to the test tube. Then, you very carefully add concentrated nitric acid, and just like before, apply some gentle heat. What's happening biochemically here is that the nitric acid is actually nitrating the aromatic ring found in amino acids like tyrosine, tryptophan, and phenylalanine. Now, once heated, you absolutely must cool the test tube down completely before proceeding to the final step, which is adding an alkali, specifically a sodium hydroxide solution. And here's how that confession plays out visually. During that initial heating phase with the nitric acid, you're going to see a distinct yellow color appear. But the true confirmation happens when you add that sodium hydroxide. That alkali causes the yellow liquid to rapidly change to a deep, rich orange. This 2-step visual shift, going from yellow to orange, is the undeniable signature that you've got aromatic amino acids out there in your solution. Section 4, the Ninhydrin Test, free aminos, revealing Ruhmann's purple. Now, this is our broadest dragnet yet, designed specifically to detect free amino acids. The protocol for the Ninhydrin test is wonderfully straightforward. You start by adding your sample solution into your test tube. Then, you simply drop in a few drops of the Ninhydrin reagent. But the absolutely crucial step here is the heat. Unlike the gentle warming we used in the previous tests, this reaction demands a boiling water bath. The Ninhydrin really needs that robust heat energy to react thoroughly with the free amino groups present in the sample. Now, notice the dramatic visual contrast we get here. For almost all standard free amino acids, that boiling process yields a magnificent deep purple or blue color, the famous Ruhmann's purple. But there's a rebel in the ranks. The amino acid proline flat out refuses to follow the rules. What's really interesting about this visual comparison is its sneaky yellow exception. Because of its unique ring structure, proline yields a yellow color instead of the standard purple. As a molecular detective, spotting that yellow immediately tells you exactly which rogue amino acid you're dealing with. Section 5. Comparing the test results. The detective's master lineup. Let's organize everything we just learned into a clear cheat sheet. Look at how brilliantly these distinct reactions separate our suspects side by side. If we want to pinpoint just tyrosine, we use Millon's test and look for that brick red color. If we're searching for the wider family of aromatic amino acids, the Xanthoproteic test, shifting from yellow to orange, is our go-to. And for a general sweep of free amino acids, the Ninhydrin test, giving us that deep purple color, unless we have our yellow rebel, proline, tells us exactly what we need to know. It's a perfectly structured system of chemical identification. All right, our final section, number 6, safety and final thoughts, best practices in the laboratory. Because look, a good detective doesn't just solve the case. They make sure they don't blow up the lab doing it. These undergraduate practicals are fascinating, for sure, but they rely on some serious chemistry, so following strict best practices is absolutely crucial. First, as we saw in the Xanthoproteic test, you must handle concentrated acids like nitric acid with extreme care. Safety gloves are totally non-negotiable. Second, avoid overheating during those water bath procedures. Too much heat can literally ruin the very color complexes you're trying to create. Finally, always start with immaculately clean test tubes. Remember, we are relying on subtle visual observations here. If a dirty test tube alters the color of your reaction, your entire investigation is compromised. So we've seen how utilizing specific chemical reagents and just a little bit of heat can completely unmask the invisible, turning clear solutions into vibrant reds, oranges, and purples to identify the fundamental proteins and amino acids that make up life. But honestly, this just scratches the surface of laboratory identification. If just 3 simple color tests can confidently reveal these invisible molecular building blocks, it really makes you wonder, what other unseen chemical mysteries are currently hiding on the shelves of your biochemistry lab, just waiting for the right detective to come along and make them visible? Thanks so much for joining me on this explainer, and keep questioning.